hozirgi dokladda ya'ni haploid olish texnologiyasi haqida metodlari, qanaqa metodlar double haploidni olishda juda ham qulay. Chunki biz o'sha gamazigota o'simliklarni olishda biz asosan klassik yo'l bilan o'sha bir 7 yilda tatishtirish orqali olamiz. Lekin aynan bu texnologiyani afzallik tarafi sizlar o'sha 1 yilda aynan gamazigotali haploid o'simlik olishingiz mumkin va albatta bunda o'sha Jochen gapiradigan texnologiya metodlar bizda hali ishlab chiqilgani yo'q. Menimcha bu narsa bizda o'sha hamkorlik o'rnatib, o'sha texnologiyani bizga olib kelish bizda imkoniyati bor. Menimcha sizlar uchun ham qiziq bo'ladi degan umiddaman. Stage is yours, Jochen, please.
Yeah, thank you, Bana, for this nice introduction. So I get used to stand here meanwhile. My today's talk is about haploid technology, which is a very powerful breeding technology and can be combined with all those things the other speakers talk about, because here we have a technology which produces true breeding material in a much shorter period of time from whatever recombinant material. I will explain in detail how it works. Haploid technology actually relies on the formation of haploid plants, and haploid plants are typically can only be obtained from haploid cells. So the cellular origin of haploid plants is always a haploid cell, and these haploid cells mainly occur in the gametophyte. We may all know with the biological background that we have these oscillations of a sporophyte and gametophyte, and the gametophyte is the hidden generation in plants which produces the gametes, which then fuse egg cell and sperm cell to produce a zygote, which is the originator cell of a new individuum. And haploid gametophytic cells we have on the male side, which is the pollen, and on the female side, which is the embryo sac. And what you can see here at the left-hand side is cultivated immature pollen. They are extracted at the unicellular stage. They are called then microspores. So these are the direct product of meiotic recombination. And at the female side, in the center of the slide, you see a gynecium, and it is indicated where the ovule resides within the structure, and the embryo sac in yellow, and the egg cell in red. And the egg cell, of course, is a haploid cell. But also the progenitor cells of the egg cell are haploid cells and could give rise to plants if we manage. And then there's a third principle of producing haploids. This is a bit complicated, coined uniparental genome elimination. That basically means after fertilization, one of the genomes of the parents is not stable in this plant and is eliminated again, leaving alone the haploid genome of one parent. And this parthenogenesis pathway can be obtained by cultivating ovaries or ovules, or by using particular mutants. And this uniparental genome elimination, dependent on which gender retains as a haploid cell, is called gynogenesis or androgenesis, and can be obtained either by interspecific crosses or by so-called haploidy inducer lines. What all these systems have in common is that you start with a haploid cell. You have to initiate cell proliferation from this haploid cell, which is difficult. For instance, if you imagine pollen, an immature pollen grain has its destination to form a mature pollen, which is typically bicellular or tricellular, or the egg cell actually stacks development as long as it is not fertilized. But by certain stress treatments or other tricks, some people have managed that. So, and once you have cell proliferation, you have a chance that you have totipotent tissue, and this can give rise to plant regeneration. And once you have this plant regeneration, during all this pathway, you need to have one event which is essential for the technology. This is whole genome duplication. Haploid plant is not capable of producing a next generation because it cannot undergo a regular meiosis, a reduction division. And therefore, this whole genome duplication is required to obtain the diploid status again. And as soon as you have this then double haploid plant, you can practically clone this plant because it's entirely homozygous. It will not segregate anymore. And the purpose for the breeder or the researcher is that what you see in this plant is what you get in the next generation. And of course, this is a breeder's dream, not to have any segregation anymore after selecting something which is useful. So now, giving you the first example, this is regeneration from the origin of immature pollen. And what you see here is a panel of developmental stages of pollen development, starting with a microspore just released from the tetrate, which is the end product of meiosis. So the microspore actually is one of these cells of the tetrate. And this undergoes a certain cellular development. It first produces a large vacuole to store nutrients for the later energy consuming process of pollen tube formation and fertilization. And it undergoes pollen mitosis 1, so a first pollen cell division. And this gives rise to two different cells, the generative cell and the vegetative cell. And the generative cell you see in the fourth picture at the bottom, which is separated at the bottom. And the vegetative cell looks very much like the former microspore. So, and right after the cell division, these two cells move apart from one another in this bicellular pollen grain. And this development goes on. But for us, for haploid technology, there's a certain window of development which is essential for us because there's a certain totipotency in these cells, and these cells can be triggered not to follow the pollen developmental pathway, but cell proliferation and maybe formation of plants. So, and this can be triggered, for instance, by putting these microspores in a medium which lacks certain nutrients, so we expose them to starvation, or we treat them with physical stress like extreme cold or extreme heat temperature. It's a border with what they can survive. So, and what you see here is the picture from the former slide where you have these microspores which have already undergone intense cell division. And when this development goes on, we get kind of microcalli. So these cells develop so much that the envelope of the pollen bursts, the exine of the pollen bursts, and then we have kind of microcalli, and these microcalli can further develop. And in the last picture, you see that they can even give rise to plants. And these are either haploid plants or double haploid plants, dependent on whether they have undergone whole genome duplication. And this basic principle, surprisingly, has been described in more than 200 different plant species, even though it does not occur in nature. So this is really surprising that it is possible to manipulate a very highly specific cell through this kind of general program of producing regenerable tissue and giving rise to plants. So, and very importantly, you have to understand the genetics of haploid technology, and this I will explain in the current slide. So here you have a representative of a hybrid plant. Different mother, different father were crossed, and these are represented by the two colors of the chromosomes. So there, I took my my beloved workhorse barley with 7 chromosome pairs, and each individual chromosome comes from a different parent, of course. And as you may all know, during meiotic recombination, they exchange fragments. So, and this is random, and this is the first situation where you have random recombination of genetic information between the two parents. And the second step of random recombination of this genetic makeup is the distribution of these recombined single chromosomes to the gametes. And you can see here 4 as representatives. These can be immature pollen grains, but also can be egg cells. So, and this is also the reason why we all look so different, because of this meiotic recombination. There's endless potential of recombination because we have these 2 steps where genetic information from 2 parents is mixed up in an unpredictable way. So, and when we are capable of developing plants from those recombined cells, then it looks like this. And as I told you, we have these plants have to undergo, or on their way, they have to undergo whole genome duplication. And then the magic happens that all in a sudden, we have all these recombinants as diploid individuals, and they are homozygous across their whole genomes. And thereby we can produce populations of recombinants, and which is very important for the breeder, because the breeder produces genetic diversity, but the genetic diversity in the ideal case is entirely true breeding. And when the breeder now selects according to the traits of these individuals something, it can be identically reproduced. And this is actually the trick of the whole technology. And I will give you an example how it could look like. Here we have crossed 2 different barleys. One has these light grains and one has these black grains. And the F1 grains look like this. But when you now grow plants from these F1 grains and use these immature pollen to produce populations of double haploids, you get this. And I was really amazed to see this for the first time in practice. Here we have produced almost 400 different double haploids, and they are all different. But the magic is whatever the grains they produce from one plant, they all look the same. So you can see here 10 examples of grains from these double haploids. They are all different. They combine different shapes, colors, and whatsoever of this grain, but one plant produces one sort of grains, and this is reproducible over generation. So, and having this culture system at our disposal, we were also interested to see the early processes of when the microspore deviates from the normal pollen development towards proliferation and to produce totipotent tissue. And here you see a regular like light microscopic recording. But when we teamed up with Michael Melzer's group, we were capable of following the development microscopically and producing little videos and see what happens. And to further empowering this technology, we used transgenic barley, which carries a GFP gene and accumulates GFP in the nucleus. So we can particularly follow the nucleus. And this looks like this. So actually, it's exactly the same object, once recorded by light microscopy and once recorded by confocal laser scanning microscopy. And we can now see the structures in the light microscope and the nuclear in the confocal. And the development looks like this. So every 3 minutes, a picture was taken over, I guess, 3 or 4 days. And what you see, some microspores just die and do nothing, but some really go on proliferating and producing a lot of cells, as you have seen in the former pictures. And this process goes on until a larger callus is formed, and from this callus we can then regenerate plants. And of course, as scientists, we were interested in individual pathways. So you see here a whole variety of things which can happen in the individual pollen grains. But we were interested to follow that up individually and also to explain what maybe is the major pathway of these structures which can give rise to plants. And here is the example of one pollen grain which follows this pathway, which ends up in plant formation or has a potential of that. And this, in contrast to normal pollen development, is features a symmetric first cell division. If you remember, I told you the first pollen mitosis gives rise to two different cells, the generative cell and the vegetative cell. And according to these culture conditions, a big fraction of these pollen grains do not produce these two types of cells. They just divide symmetrically, giving rise to two same cell types. And of course, you need to look carefully. What you see here is one nucleus of the microspore, and this smaller green structure is the pore, the preformed pore of the pollen grain, where later the pollen tube comes out. And you should not confuse these two. So here it is about the upper pollen grain and the larger nucleus. And when we look according to the development, you see the nucleus moves within the cell and all in a sudden divides in two nuclei, which have the same size, the same shape. And this proliferation now goes on simultaneously. They divide again. And so it goes on producing this multicellular structure. And when we counted the events within our culture, we came up with a little bit over 50% of all pollen grains following exactly this pathway. And altogether, we found 9 different pathways. At least 2 of them we assume can give rise to plants. But I will also show you another example, which also unfortunately happens quite often, that these cells do not survive the culture. And here, this pollen grain in the middle tries a lot to undergo cell division. And it takes a lot of time that something happens. But when something happens, you will see the whole cell messes up. And this we call programmed cell death. And you will see a burst of the nucleus forming lots of micronuclei. And the cell is no longer capable of dividing and surviving. And in doing so, we maybe coincidentally came across the observation of a very interesting phenomenon. And this is how the genome duplication can occur in this process. You may have heard about colchicine treatment so that you can obtain higher haploids. But here we have the phenomenon that a fraction of the plants you produce are already have undergone whole genome duplication. In barley, that means they're diploid. And here we see the phenomenon which can give rise to this genome duplication when you look at the 2 nuclei with it. So here you see all nuclei in one pollen, which has an advanced stage of proliferation. And in the right corner below, you see a pair of nuclei, and you have to follow what they do. So, and together with light microscopic observations, we found that the cell wall between 2 newly formed cells is often not perfectly closed. And when these nuclei can get in touch with one another of these 2 newly formed cells, they fuse. And this is exactly what you see. And to show it one more time in a close-up, here you see these 2 guys again, how they get in touch and fuse to one diploid nucleus. And this is, of course, for us working in this field, a very exciting moment to see that. marriage of plant nuclei. Yeah, and when we look closer, so these events can happen all over the time. So that can happen right after the first division or later. And then also chimeric plants can be produced when the later the nuclear fusion occurs. Of course, only a part or a sector of the plant is diploid, but you will see that because the diploid sector will give rise to seeds or grains, and the haploid part will not produce any. So now I come to another principle of forming haploids, and this is called uniparental genome elimination. Here you see also as representatives of parents only the egg cell and the sperm cell, and in certain constitutions of interspecific crosses or certain mutants which have the capacity to act as a haploid inducer, these hybrids are produced. And then we see the phenomenon that the genome of one parent is eliminated again. And there are different mechanisms where maybe roughly saying the management of the two genomes does not correspond very well with each other, so that one genome accidentally gets lost. But of course, we have the benefit produce haploid cells or haploid plants by that, because if only one genome is left, we get a haploid cell plant. And again, here we have to make sure that whole genome duplication is undergone, and then we will have double haploid plants. So, and those examples here are well known. So if you cross barley with its wild relative, Hordeum bulbosum, you will see this effect in a couple of events. When you pollinate wheat by maize, you will see this principle. And there are several other, maybe not so well-known examples. One of them is that it also works with potato when you use a certain wild species as a pollinator. But there's the other group of intraspecific crosses. So pollinators of the same species with a certain genetic constitution are also capable of serving as those haploid inducers. And as such a system is heavily used within maize, it was also the first gene which was identified to be responsible for that. And this is a phospholipase, PLA1, given in red. And this PLA1 is mutated, and therefore this is the reason that the male genome is eliminated after fertilization. And also centromeric proteins can be responsible for this effect. The centromeric histones H3, when it's modified, this was found in Arabidopsis that it works. And in Arabidopsis, interestingly, you can use these mutants either as a paternal parent or also as a maternal parent, and it goes in both directions. So, and since this gene was identified, of course, we were looking whether there are orthologs in other species, and we found an ortholog of this PLA1 also in barley. And I will not stress you with the details given here, because I have many other things to tell. There's a second gene which also can contribute to that, but this is not of interest today. What we did, we mutated PLA1 by targeted mutagenesis using Cas nuclease technology, as I presented yesterday, and we produced quite a number of those mutants, and 70% of the transgenics produced were mutated. And here you see where we selected target motifs within this gene, which comprises 4 exons. And below you see, again, the same principle. Upper line is a wild type, and below there are different mutations we obtained. And as you can see, smaller deletions, insertions of one nucleotide, larger deletions, quite a panel of different things we got. So, and we rendered those mutants homozygous and then pollinated. First, we selfed by reproduction, and then we developed a method using these haploidy inducers to produce haploids in any barley or from any barley. So, and the method is that we have to emasculate and pollinate the maternal parent. And 7 days after manual pollination, it looks like this. These grains have already developed to some size. And since the endosperm is kind of handicapped, we need to rescue these embryos from collected caryopsis at that stage. But the embryo rescue works pretty well on a suitable medium. So, and when we selfed these plants, we already got some haploid plants. This was indicative of the method may already work. But of course, for research and breeding, it would be much more interesting to pollinate another accession with the haploidy inducers. And here in the diagram, you see the efficiency, what is the percentage, or what is the percentage of haploid plants amongst the plants you produce after such a pollination. And we started at the right side with Golden Promise, which is a selfing thing because we did it in Golden Promise. And this was about 6% of the plants were haploid. And then we used the two former reference genome accessions, Morix and Barker. And in Morix, we found a similar efficiency. And in Barker, surprisingly, it was much higher. It was 16%. And these are efficiencies a breeder can really work with. So, but we want to get one step further using this technology also for genome editing. And what I present here is a very specific approach where we transformed the haploidy inducer with a construct carrying Cas endonuclease and guide RNA. So in this case, we transformed maize with such a construct with a guide RNase to be specific for a wheat gene. So they do not harm in maize at all. They do not find a target, and you can produce this maize, but you can pollinate wheat with this maize, and this is what we did. So you see here in the sperm cell, which is with this yellow interior, there's indicated the construct, the transgene construct inserted. And when we now pollinate wheat by this maize, this Cas endonuclease guide RNA all of a sudden has a target in the cell. And of course, the big question was, does it find it? And the concept was then that using this haploid technology, we know there's uniparental genome elimination. The parental genome will be eliminated after a short period of time where these two genomes were in one cell. So, and then of course, we would need to make sure there is whole genome duplication, and then we would have mutants which have no transgene anymore, or right away no transgene. So, and but this is not the major thing because these transgenes can be crossed out easily. But what for me was much more important is that that wheat transformation is pretty genome dependent. So we are not free in selecting wheat genotypes to be manipulated. And this haploid technology is much less genotype dependent. And therefore, this was actually my driving force, except that it's a cool approach to look whether we can get it. And one other thing is once you have such a haploidy inducer carrying this specific Cas endonuclease and guide RNA, you can trigger specific mutations in a whole panel of different genotypes with this one plant even. So, and there was one concern when you imagine the different sizes of egg cell and sperm cell, egg cell coming from wheat, sperm cell coming from maize, that the sperm cell has not enough substance to bring in this endonuclease. And here I show you the dimensions of these two cells. So in my former scientific life, I prepared gametes of cereals. And what you see here is a pollen grain put in a mannitol solution, bursting because there is high osmotic stress, and it releases the two sperm cells, which is indicated above. And clever enough, we used transgenic material, GFP expressing material, so that we can see these two sperm in a confocal very clearly. And this told me that we can express Cas endonuclease efficiently also in sperm cells by using a ubiquitin promoter. So next question is the sperm cell big enough to bring in the active agent? And here I show you another part of my former life when I isolated those gametes. I also did in vitro fertilization, and here you clearly see on the right-hand side, the egg cell is a huge cell, and the sperm cell is actually a dwarf in comparison. And by the way, because it's also very exciting and it's also about haploid cells, I show you what we did here as a little side story. We were capable in an electric field to fuse these two cells with a little pulse of alternate current. And you see within a second, you see that these plasma membranes fuse in the middle, and in the right-hand side, you see the remnant of the sperm cell almost taken up by the egg cell, giving rise to a zygote. So, and the question whether the sperm cell is big enough or accumulates enough Cas enzyme was answered then by our experiments, where we then looked into the plants we generated, whether they have mutations. And in the beginning, it didn't work, but we worked hard, and in the end, we found some. And this is pretty busy, and I have to explain a little bit. It's always the same principle. The first lines are also always wild-type, but here are shown several groups of events. So we did it in common wheat, and we used two different genes, both related to the semi-dwarf trait I reported yesterday. So it is the brasinosteroid insensitive gene we mutated, and the semi-dwarf 1 gene, which is gibberellic acid oxidase gene, which also contributes to plant height. And as you can see, we did get mutations in common wheat and in durum wheat. And we, in all 5 genotypes we used, we got mutations, which is also a good sign that genotype dependence is not all too high. We got mutations in all subgenomes of wheat, A, B, and D, and we used 2 different genes and 3 different target motifs, and all gave us examples where it works. And this state of development was enough then to have a good publication about it, and to talk with colleagues where we could apply it. With that, I would like to conclude my talk, and thanks for your patience and attention.
Thank you, Jochen, for the wonderful talk. Ozgina o'zbekchada bularni anaqalarni aytib o'taman. Hozir o'sha bitta kichkinagina hujayradan, ya'ni chang ichidan o'sha o'simlik olishini gapirdilar. Bu juda ham ajoyib texnologiya. Yana bitta tarafi mikroskop bizada umuman yaxshi rivojlanmagan. Mikroskop bizada umuman anaqasi yo'q, texnologiyaga kirib kelmagan. Aynan mikroskop orqali biz juda ko'p narsalarni aniqlashimiz mumkin. Kimdirdir o'sha mikroskopik ishlar bilan qiziqishiga taklif qilganda, yo'q, bizada mikroskop umuman qiziqmas, hech kimni mikroskopik ish bilan ish qilishni umuman xohlashmagan. Lekin bu o'sha ko'rayotgan o'sha prezentatsiyada aynan mikroskopni kuchi juda ham kattaligini, bizga juda ko'p ma'lumot berishligini ko'ryapmiz, to'g'rimi? Keyingi anaqasida aynan eng qizig'i o'sha ikki xil turdagi o'simlik, masalan, bug'doy bilan makkajihonni chachishtirib, aynan oxirida bitta bug'doy o'simligini olish ham ajoyib texnologiya. Are there any questions? So,
Sorry, my English legal. Hozircha tarjima qilasiz. Judayam zo'r prezentatsiya uchun tashakkur. Avvalo, bubble gaploid texnologiyasini paxtachilikda qo'llash bo'yicha savolim bor edi. O'zbekistondagi iqtisodiy ahamiyatga ega bo'lgan ikkinchi ekin g'o'za. Bugungi kunda yangi navlar yaratishga O'zbekiston paxtachiligi muhtoj, lekin bu uzoq 10-12 yilni talab qiladi traditsion metodika bo'yicha. Bubble gaploidni ustada, g'o'zada qo'llashda nima muammolar bor va ularda yechimlar bormi? Xuddi hozir bug'doyda yoki makkajo'xorida qo'llagandek qo'llasa bo'ladimi? Yana mikrofon olmay, ikkinchi savol. Kas-9 edit bo'yicha target to'g'risida gapishmadi. Off target holatlarida yechimi topilganmi? 2024-yilda ham o'sha kerak emas joyda kesib yuborish holatlari yoki bunaqangi решение qilingan.
I did not understand a single word, but it was a pretty long question.
I will translate. So in our country, the second important crop is cotton. So it's possible to do this, play this method to cotton. This question, first question, and if yes or not, in how?
Okay. I cannot promise. I didn't look into the literature where are already examples for cotton. That might well be, but maybe not. As I gave you the number, and the number is old. So I guess from the 90s, there were already 2,000 different plant species in which the regeneration from isolated microspores or answers containing those microspores give rise to haploid or double haploid plants. So the chances are pretty good. And I can also give you maybe another hope. We are currently working in several species which have not been that important in history, but now become more important, like legumes or pseudocereals. And for all those, there is no such technology available. But having three choices, using microspores or parthenogenesis, ovioculture, ovariculture, or finding another related species, when you pollinate, you may get haploids. You have lots of chances, and this is what we do. So we don't know whether we are successful, but we know the principle is pretty universal, and therefore it's always good to give it a chance, work hard, and maybe you will be the successful one.
Men aynan bu texnologiya bilan ishlagan olim sifatida ayta olishim mumkin. Bu narsa mumkin har bir o'simlikda, faqat savol biz o'sha to'g'ri optimal bo'lgan condition sharoitni yaratib bersakkina u o'sha bittagina kichkina tiny single hujayradan bitta o'simlik olish mumkin. Buni iloji bor. I will he has a second question was
maybe one comment.
Yes.
Since I know that Uzbek people are pretty smart and hard workers, I have a Uzbek guy working exactly on this in the pseudocereal quinoa, which has been an orphan crop, but now gets a lot of importance. And if we want to unlock this species for modern breeding, we need haploid technology. And Mohammadian is working exactly on that in quinoa.
His second question was, hozirgi kunda o'sha eng qiyin, endi nima desam, muhim bu ahamiyat kasb etayotgan o'simliklar bor. Oldin umuman e'tiborda bo'lmagan, hozirgi kunda juda e'tibor talab qilyapti, chunki ularda oqsillar ko'pligi yoki qaysidir ma'noda oziq-ovqatda o'sha nima desam, bir o'zida kerakli anaqa vitaminlarni tutgan o'simlik bo'lgancha, hozirgi kunda unda o'sha gen injeneriyasi yoki boshqa anaqa texnologiyalar qo'llayapti. Bizada o'sha Muhammadjon Mirzaahmedov, ya'ni Center genomikada ishlagan, hozirgi kunda quinoa o'simligi ustida aynan double haploid texnologiyasini yo'lga qo'yish va transformatsiyasini yo'lga qo'yish bo'yicha ishlayapti.
Quinoa.
Yeah, no. The second question was but it's the question actually not related to directly double haploid technology. This was related to genome editing. Like, how about off-target analysis? Is there any kind of solution? Actually, how we can come up with this off-target effect?
Off-target mutations are mainly seen as a problem. I do not see that as a problem. Maybe a safety problem, not. Maybe for breeding, it can be a problem if you have an unexpected effect, which is deleterious. Of course, we need to care on that. But off-target mutations produce additional genetic diversity. And we have experienced, in cooperation with Sophia, a very nice example where we mutated the nudum genes to produce naked barley, as reported yesterday. And we have off-target mutations. And in the beginning, we did not really care on that because I thought, okay, there are mutations all of the time. So why we should care on these specific off-target mutations? But by then carefully looking at that, we found another family member of this gene mutated, and this initiated us to have an entirely new story on Vex formation at the plant surface. And this is for me a very good example. So there is no safety issue because there is always spontaneous mutagenesis in all organisms. And of course, this has a certain risk, but when you have off targets, there is no reason to regard these as more dangerous as any random mutation. And random mutations appear to happen much more often than off targets appear to happen. And therefore, the risk is not even comparable. It's like a drop in the ocean, you know? And I see it much more as a chance that we create additional biodiversity, which we can use in science or even in breeding. But of course, I have changed my mind. We should not disregard these off-target mutations. We should look at them, maybe to profit the best way out of them.
Off target analiz bo'yicha asosan aynan yaqinda o'zimni o'sha maqolamda qilganman. Siz o'sha guide RNKlarni tanlayotganingizda genni siz o'sha tanlangan guide RNKni tekshirib ko'rish kerak. Juda boshqa genni o'chirib qo'ymaslik, ya'ni boshqa joyga to'g'ri tushib qolmasligini oldin tekshirish kerak. O'shanda agar sizda hech qanaqa genga o'tirmayaptimi, demak sizda hech qanaqa off target mutation bo'lmaydi. Lekin men nima qilganman, birinchi уже transformatsiya qilib, уже menda mutatsiya bo'lgandan keyin men off target analiz qilganman. O'shanda men ko'ryapman, men boshqa genni ham o'chirib qo'yganman. Qaysidir ma'noda to'liq emas, qanaqadir 70% bu yerda ya'ni functional bo'lgan tarafi ham bor. Agar domla aytyaptilar-ku, siz o'sha bilmagan holda boshqa genni o'chirib qo'yib, qaysidir ma'noda foyda olishingiz ham mumkin. Lekin buni baribir ko'zdan qochirmaslik kerak. So thanks so much, Jochen, for the interesting discussion. Now I think, sorry, Jochen, so.
Assalomu alaykum. Hurmatli anjuman qatnashchilar. Barnopa boyagi so'zimizga to'ldirib ketaman. Bizda mikroskopiya tadqiqotlar yaxshi rivojlangan. G'o'zada asosan rivojlangan, bug'doyda u qadar emas. Ya'ni genetik tadqiqotlar demak dunyoda ikkinchi o'rinda turadi. Bizda demak tadqiqotlar Milliy universitetda genetik kolleksiya bor G'o'zani. Sanamyan va uning shogirdlari tomonidan yaratilgan. Hozirgi shu genetik va molekulyar genetik tadqiqotlarni birlashtirgan holatda biz ham Haploid texnologiyasi yaratganmiz va oldik va Xilzotum turida birinchi marotaba dunyoda Haploid oldik. 5 ta Haploid o'simligimiz bor, har xil demak boshlang'ich materiallar asosida va 2 ta demak double Haploid liniya hozir olganmiz. Bundan tashqari yana o'sha genetikani yutug'i biz respublikamizda, xususan biz laboratoriyamizda biz o'sha genetik kolleksiyadan unumli foydalangan holatda demak yangi xromosomasi almashgan liniyalar yaratganmiz. Bu AQSh kolleksiyasida mavjud emas. Shuning uchun men sizga taklifim kelgusida aynan qaysi o'simlik ekanligini fiksatsiya qilib o'tsangiz yaxshi bo'ladi. Bug'doyda u qadar rivojlanmagan. Mikroskopik tadqiqot, lekin G'o'zada yaxshigina rivojlangan. Mani savolim bordi bir necha. O'zbekcha beraveram. Birinchisi ruscha tushunmaydi. Birinchisi qaysi metodlar qulay? Ya'ni Haploid texnologiyani olish uchun birinchi bu spontan, tabiiy ravishda, ikkinchisi sun'iy ravishda turlararo, turkumlararo chashtirish Haploidlar olish va uchinchisi siz aytganda changchilar yordamida Haploid o'simliklar olish. Lekin bu metod hamma o'simlik uchun qulay emas. Xususan g'o'za uchun, ya'ni changchilar orqali olimlar tomonidan bir necha tadqiqotlar amalga oshirilgan, lekin muvaffaqiyatli chiqmagan. Changchi yordamida Haploid olish. Birinchisi
to'liq
uchta metodda qaysi biri qulayligini aytib bersanglar bo'lsa.
Jochen, what you described in your on your talk, so there are like several methods to get to obtain the double Haploid, right? And in cotton, which method might be or can be suitable?
This is not predictable, but my advice is to start with this pollen, because you have so many pollen in most of the plants that the chance is good that at least a few of them decide to follow this pathway in contrast to normal pollen development. And in history, actually, the majority of all viable methods are based on this principle. And only if you fail with it, you have these alternative things with parthenogenesis and genome elimination. First is always microspore culture, answer culture, following the principle of pollen development.
Thank you. İkinci savolim как раз elimination of chromosomes. Bu yerda bu savolim esa Pershini uchta stadiyaga bo'lgan. Eliminatsiya xromosomasining genom, elimination genom, ikkinchisi ya'ni частичная eliminatsiya, ya'ni bir necha xromosomlar yo'qolib qolishi, uchinchisi alohida xromosomlar eliminatsiyaga uchrashi. Bunga nima sababli bo'ladi? Ya'ni H3 oqsildan tashqari.
The question is
Elimination.
the elimination of elimination of chromosomes, and then how this elimination happens. Sometimes the one chromosome eliminated, or at the same time, or multiple chromosomes eliminated, and what could be the reason for the elimination, and all different types of elimination.
Okay. Yeah, this is pretty complicated, and therefore I did not want to bother you with that, but I try to make it short. So there are different mechanisms that one parent's chromosomes are eliminated. And as I tried to generalize is that the chromosomes of both parents are processed in one cell simultaneously. And as these chromosomes come from different species, the processing and reshaping and undergoing cell cycle and so on is not simultaneous when you have a hybrid. And this is also a reason why in many crosses no hybrids can be obtained, because one genome is eliminated and then the genome of one parent is left alone and you have no hybrid. So one more particular mechanism is when a chromosome undergoes development, it forms an active centromere, which can be taken by the mitotic spindle to be drawn away as a chromatid from the other to be placed to one daughter cell and to the other. And the centromere is not formed or still not mature enough in one parent's chromosomes. And so these chromosomes are not allocated to a daughter cell. And this is a mechanism which is proven. But we guess there are also other things playing a role, but this is just an example how it can happen.
Thank you. Oxirgi savolim, ya'ni bu ham Haploid bo'yicha xitoyliklar o'sha CRISPR-Cas texnologiyasidan foydalangan holatda DMP genlarini o'chirganda va Haploid o'simlik oldi. Ya'ni g'o'zani gasev xizotini turdi. Ammo bu genlarni o'rgansam, bu genlar oilasi g'o'zada qurg'oqchilikka ham javob berar ekan. Hozirgi kunda g'o'zada qurg'oqchilik bu ayni aktual muammo. Haploid texnologiyasidan olish, ya'ni buni asosiy maqsadi biz double haploid olamiz va seleksiyaga qo'llaymiz. Seleksiyada qo'llaganki qurg'oqchilik chidamsiz qolsa, u qanday nav yaratsa bo'ladi? DMP genini kirib qo'ydi. DMP prezentatsiyada ham ko'rsatdilar.
There are gene which called DMP. It is responsible to induce the double haploid, and if he said when he searched the gene, it belongs to drought-related.
It's a whole gene family.
Yeah.
It's difficult to spot the most related ortholog. Yeah, I agree.
Like switch the function of the gene, and we have a double haploid plant, but we lost the drought tolerance. And what is the kind of advantages of doing this? So because drought is also really kind of main problem in our country because of the dry condition. So
I don't understand the question. When you have a haploidy inducer, and this haploidy inducer is mutated in PLA1 or DMP, then this genome is not included in the plant you produce. So this is exactly the mechanism that the genome of the haploidy inducer is eliminated. So all these genetics do not play a role in the double haploids produced. So you can mutate whatever you want in this inducer line because it doesn't contribute to the genetic makeup of the plants you produce.
Tushunarli. Thank you.
Yeah, thanks so
Thank
much.
you for your interest.
Yeah. Now time to break. So we have a coffee break now. Then we will continue after the coffee break.